বুদ্ধিও বহুত। মই চবকে দিলো নহয়। এতিয়া তাৰিখ মানে একদম
হেল্ল' পেমিলি।
ছাৰ
ঠিক আছে, ভিডিঅ' দিয়ক।
ছাৰ শুনি পাইছো ছাৰ।
ঠিক আছে, ঠিক আছে, ঠিক আছে। ৰিচোৰ্চ পাৰ্চন জইন কৰিলে নেকি?
ছাৰ জইন কৰিছে, এতিয়া কহজ বনাই আছো ছাৰ। উইদিন এ মিনিট উইল ষ্টাৰ্ট দ্য প্ৰগ্ৰাম।
অ'কে, অ'কে, অ'কে।
ছাৰ আমি ষ্টাৰ্ট কৰিব পাৰো এতিয়া, আমাৰ স্পিকাৰো জইন কৰিলে। ছাৰ shall we
Okay,
start the
start.
program?
Yes, you can. Yes, you can.
Yes, sir. Good evening and warm greetings. I take the honor and pleasure to extend a warm and hearty welcome to everyone. Respected Principal, Dr. Sabyasachi Bahunda Sir, Vice Principal Ma'am of Gorgan College, Dr. Rina Hendik, IQAC Coordinator, Dr. Surjit Sakya Sir, respected faculty members of Gorgan College, and my dear participants. We are honored to have such a distinguished resource person, Dr. Ramjan Ahmad, joining us today from Thailand in this second day of the invited lectures on biological sciences. On behalf of the organizing committee, I take the pleasure to welcome you all in this second day of the two-day invited lectures on biological sciences, or which is organized by the Department of Zoology and IQAC Gorgaon College, Himuluguri, Assam, India. To begin with the workshop, I would like to request our eminent academician and principal of our college, Dr. Sabyasachi Mahanta sir, to present a welcome address. Sir, please do the honor.
Thank you so much, Dr. Pemili Lanthasa. A very good evening to you all. A very good evening to you all. Dr. Rina Hendik, Vice Principal and HOD of the Department of Zoology, Gorgaon College. Dr. Pemili Lanthasa, Coordinator of this invited lecture organized on biological sciences. Assistant Professor, Department of Zoology, Dr. Surojit Soikia, Coordinator IQAC of Gorgaon College. Today's resource person, Dr. Ramzan Ahmed. We are very glad to know that he has been joining from Bangkok, Thailand. Respected faculty members and my dear students and participants. It is indeed a great pleasure of us that we have been organizing the Department of Zoology in collaboration with IQAC Gorgaon College has been organizing first type of events, invited lectures. Today is the second day. This is the second program, invited lecture on biological sciences. And the organizers have invited Dr. Ramzan Ahmed, an eminent researcher. He is a postdoctoral fellow in the Department of Transfusion Medicine and Clinical Microbiology, and also a faculty of Allied Health Sciences of Chulalongkorn University, Bangkok, Thailand. I offer my sincere thanks and gratitude to the resource person, and I welcome the resource person. And I also welcome all the participants, and I appreciate the endeavor and initiative that has been taken by the organizers to organize such an event on biological sciences so that our students can be mostly benefited. Not only the students, our teachers will also be benefited from this type of events. And the Gorgaon College has been relentlessly working hard to give something, to impart some special education, some specialized education, expertise education to the students so that our student can acquire the skill of problem solving, acquire the skill of critical thinking, and they would be able to acquire practical knowledge in their respective fields. And the today's lecture would be on construction and interpretation of phylogenetic trees that would be delivered by our resource person, Dr. Ramzan Ahmed. I hope the session, the presentation that would be made by Dr. Ramzan Ahmed and the deliberation that would be taken place in this session will definitely be beneficial for the students as well as for our teachers and research scholars. So with these few words, I hope the event, the program would be a big success. And with these few words, on behalf of the Gorgaon College fraternity, I welcome our today's resource person, Dr. Ramzan Ahmed, and I welcome all the participants. And I appreciate the initiative of our organizers. And with these few words, wish you all the very best. And I hope the program would be a big success. And with this few words, I declare the session open. Thank you so much.
Thank you, sir. Now, I would like to request our Vice Principal and Head, Department of Zoology, Gorgang College, to say a few words. Over to you, ma'am.
Thank you. Good afternoon to you all. On behalf of the Department of Zoology, Gorgang College, I welcome our respected Principal Sir, Dr. Sabyasachi Mahanta, respected IQAC Coordinator, Dr. Surajit Saikia, Dr. Ramzan Ahmed, esteemed resource person, respected faculty members, and dear participants and students. Welcome. I welcome you all to the invited lecture on biological science organized by the Department of Zoology, Gorgang College, in collaboration with IQAC. In the series of invited lecture, we intend to bolster the aptitude of the coming of age students towards study and research of various critical aspects of biological science, respect of biological science or biology. In today's lecture, Dr. Ahmed will share insight on the matter of construction and interpretation of phylogenetic trees, which is a growing prospect in the light of enhancing biological biodiversity during climate crisis. I hope that all the participants of the lecture will substantially benefited from the session enriched by Dr. Ahmed. I wish you all the very best, and I also wish the overall success of the program. With these few words, I thank you all. Thank you very much.
Thank you, ma'am. Now, without much delay, before starting the session, I would like to introduce our resource person to the participants. Dr. Ramzan Ahmed is currently working as a postdoctoral fellow at Transfusion Medicine and Clinical Microbiology. He is also a faculty of Allied Health Sciences, Chulalongkorn University, Thailand. He did his PhD in microbiology from Assam University, Silchar. His area of interest covers microbiology, molecular biology, malaria, antimicrobial drug resistance, food microbiology, parasitology. He published his research paper in many national and international journals. With this, I would like to hand over the session to our today's speaker. Dr. Ahmed, over to you.
Okay, thank you so much, everyone, especially the principal sirs and Emily madam. So thank you for the giving me the opportunity. So I will start with today I will provide some information about the how to construct the phylogenetic tree. Am I audible?
Yes.
Okay. So today I will discuss about the construction and interpretation of phylogenetic tree. Before going to discuss about the phylogenetic tree, how to construct it. I will like to share the what is phylogenetic tree and what is the purpose of the phylogenetic tree. Basically, what the phylogenetic tree we do that is it's a graphical representation among the organism. Okay, so basically, how means that a phylogenetic tree means that that evolutionary relationship among the organisms or a species or group of organism are known as phylogenetic tree. So in that case, how we can say that the phylogenetic tree means that how they are related. Suppose one organism to another organism, how they are related, or suppose that in human being, so how we related to our ancestors. So not only the human being, also many other organisms also, or species or particular or up to the genomic level also. So in simply we can say as like the representation path between common ancestors to divergent descendants. So divergent descendants means how can we say that? So how we differ from that our ancestors to the present day organisms. So first, that phylogenetic terms that comes from the Greek word the phylon, and race means the race or the lineage. Lineage means the linkage of that organism from one organism to the another organism, or how they are related. And the genesis means that origin. So that origin of particular organisms, how the organisms are originated or during the period of evolutions. So in our earth, so there are so many organisms are there. So they are maybe that genetically different, or if we see about the genetic informations or the DNA sequences. So one organism to another organism, they have different they have a variation in their genomes or the DNA sequences. Okay. So, or whether it is genetically or the phenotypically means that visible characteristics, how the from one organism to the another organisms, the visible characteristics, whether they are similar, whether they are the dissimilar. Then next is that a type of evolutionary tree. So when we construct the tree, so there are 4 different types of tree are there: the rooted tree, then unrooted tree, bifurcating tree, and multifurcating tree. So in that way we can construct different type of, or we can say the different forms of tree. When we construct the tree, so there are different forms of tree are there. So what is that rooted tree means? They have a mainly the rooted tree is the described phylogenetic tree sharing the common ancestors on one node. So in that figure you can see here that so they in that rooted tree, so they have a common node is there. This one is the common node. Later I will show you that what is the node, common node, and they have a common node. But in case of that unrooted tree, they have no any common node or the root is there. So in bifurcating tree, what is bifurcating tree? In bifurcating tree has 2 branches. Okay, you can see here also the bifurcating tree. So one branch is only the single one organism, and another branches is that only here is the 2 organisms are connected in that single branch or the single node, we can say at that is the bifurcating tree. And multifurcating tree, that means the multiple branches on a single node. You can see here also that in multiple branches on the single node. Here you can see here is one branch, here is one branch, and here is the another branch. So these are the 4 evolutionary tree we can use in the during the construction of the phylogenetic tree. So before we construct the phylogenetic tree, so few terms are there we should know about it, that the phylogeny, then common ancestors, taxons, taxa, node, then sister group, outgroup, and clade. So these few terms are generally when we interpret the phylogenetic tree, you can see here also that when we interpret the phylogenetic tree, so we should know these terms like common ancestor, what is common ancestors, taxons, taxa, node, sister groups, and outgroups, and clade. So phylogeny, we know about the phylogenetic method of construction of the phylogenetic tree. The common ancestor, the common ancestor means that a group of organisms sharing the common features with the descendants. That means we generally say that, so how the present day, suppose human being are connected to their ancestors. Okay, so not only the human beings, also any other organisms, also whether it is microbes or that animals or the plants. Okay, how they are related to their ancestor or their. And taxons means an organism of their entire taxa. Taxons means the organisms of entire taxa. So in taxa means that group of organisms. So taxa means that group of organisms means, for example, like group of organisms like that animals, like for example, cow or the buffalo or the goat. So a taxa, that is a group of organisms from a species or from a different species. Whether they are the taxa is that they may be the same species or the different species. And nodes represent the common ancestor of different taxons. So here is the node. You can see here is the node is the common ancestor of the different taxons, means the different, whether the same type of species or the different species in the same node. So or we generally say as that this node is called as the common ancestors. And then sister groups, the sister group is the two or more taxons share the same node. So for example, like taxon 1 and taxon 2. So these two are the sister group, or these two are the sister groups. They have a common node or the common ancestors. So you can see here that taxon 3, the taxon 3 means this is the outer group, or they have no so connected, they are not connected with the like node 1. So the taxon 3 is connected to the node 2. So these are the example of the sister group. Then outgroup also similar, like the taxon is outside the interest of the group other than the common ancestors. So like other than the common ancestor, like here is the node 2. You generally see is that node 2 is there and node 1. So they are connected on the same node. Okay, but in case of like taxon 4, so they are totally different. Connected is that taxon 4 is connected to the node 3. So taxon means the group of organisms, whether that is the animal or the human beings or the microorganisms or the plants or any other organisms maybe. So this is all about that taxon is, that means taxon 4 is that outgroup. We generally say, like for example, like say if we take that for example as a taxon 1 or taxon 2 or taxon 3, for example, we take that human beings. Okay, and taxon 4 outer group, we should take that like as an example of animal like whether it is cow or buffalo or any other animal. Clade. A clade is a group of all organisms from a common ancestor. So that means clade is a group of all organisms from a. So for example, like that this one node 2. That node 2 is that connected to the taxon 3 and taxon 2, taxon 1 or node 1 also. So they have a common ancestors in the node 2. So in this figure, we generally say like taxon 1, taxon 2, taxon 3. So we can see, we can also call it as a tip or leaf of branches. Tip or leaf of branches. And taxon 3 is out group to taxon 1 and 2. But taxon 1, 2 have similar node, means they have a common ancestors. Okay, but taxon 3 have also that another node is there, but they have also both this taxon 3 and 1, 2. They are connected in the same common ancestor, but not in the same group. Okay, but they have some similarities there. Because of that, some similarities, that that node 1 and the taxon 3 is connected to the same node, that is node 2. So here you can see that in terms of phenotype or genetic sequences, while the organisms of taxon 3 is not so similar because it is not the same lineage or the node, though all three are sharing the common ancestor on node 2. So however, that means taxon 3 or taxon 2 or taxon 1, they have share some common characteristics. Because of that, some sharing the common characteristics, they are connected to their ancestors, that means node 2. So here you can see that the ancestors and present day species or present day any other organisms, how they are connected. Suppose this one is the node or branch point. We generally say the branch point. These are the branches, or these are the taxons like A, B, C, D, or E. So E is the outer group. So next is that construction of phylogenetic tree. How we can construct the phylogenetic trees. So in construction of phylogenetic tree, first of all, we should know about the organisms or the particular organisms or a gene family. The gene family means in particular organism have their genomes. In that genomes, there are many different type of genes are present. So particular group of genes we can target, whether it is particular group of genes, or for example, like we can target for in case of bacteria like 16S ribosomal DNA, or in case of eukaryotic organisms like 18S ribosomal DNA. For the 18S ribosomal DNA and 16S ribosomal DNA for the bacteria, for that means these genes are particularly identify the particular species, a bacterial species, 16S ribosomal DNA. So in case of like eukaryotic organisms, so like human or animals, or so we directly target the 18S ribosomal DNA for in case of eukaryotic organisms. So choosing the appropriate molecular markers. So molecular markers means that, say, is a short sequence of DNA. So maybe that short sequence of DNA means that whether you choose that particular gene or the particular gene of interest. In case of like bacteria, we choose that 16S ribosomal DNA, or we can choose any other particular genes also. Then after that selection of your target, you have to do the PCR amplification. You have to do the PCR and have to do the sequencing. After sequencing, you have to do the, after sequencing, you will get the DNA sequences. So DNA sequences, I'll show you the DNA sequences. So these are the DNA sequences. For example, these are the DNA sequences. For this, I have for the during for construction of phylogenetic tree, I choose like E. coli 16S ribosomal DNA. This sequence is a 16S ribosomal DNA sequence. So after sequencing, after getting the DNA sequences, we have to do the sequence alignment. So sequence alignment means that we suppose we have like 20 or 4 different organisms are there. Okay, so we have PCR amplified. Okay, PCR amplification, then and sequencing of the 4 or maybe that more than 4 organisms of the. Okay, so and we have to take also that during construction we have to take also the another outer group also, not only the same group, but outer group sequence we can get it from the website, NCBI website. So after that getting the sequence, you have to do the multiple sequence alignment among the sequences, among the different organism sequences, and then you have to do choose the evolutionary model. Okay, so in during construction of tree, so there are many that model is there, like Kimura model or any other model is there. So you have to choose according to your experiment. Then phylogenetic tree construction. Then you have to do construct the phylogenetic tree. After construction of the phylogenetic tree, after when you did the phylogenetic tree, and then you will get the some graphical representation after construction. And then after construction, you will evaluate or interpret the phylogenetic tree. Basically, the phylogenetic tree, how to suppose one organism, how they are related to the one organism, the another organisms, but how much characteristics they have shared among the organisms. So basically, we should know even that during evaluation, so we can know the how much they are related to one organism to the another organisms. So let's go. So for construction of phylogenetic tree, so we can use whether it is online based or maybe some other different type of some software is there. So generally, I choose the MEGA. So that is molecular evolutionary genetic analysis. This is the free software, so we can get it from the online. So for construction of phylogenetic tree, we can use that MEGA, or we can use also different other tools also is there online. That is iTool also is there, so we can get it for online. We can do it for before that construction of the phylogenetic tree. You should know the sequences. Okay, you should after PCR amplification, you get the sequence. Okay, so that sequence should be in the FASTA format. Okay, so that means that go to the National Center for Biotechnology Information. For example, if we don't have a sequence, so that center, that means this one is a database, that National Center for Biotechnology Information. In that database, so worldwide they have deposited the DNA sequences of any organism, so that is human or bacteria, fungus, okay, any microbes or any plant organisms, plants. So in that website, we can easily get the DNA sequences. Not only the DNA sequences, we can get the protein sequences also. We can get the amino acid sequences. Okay, so after download the sequences, or you can copy or paste in a notepad file, and that file should be named as a, for example, I choose that one is XYZ or any name, and .FASTA should be that .FASTA, and save it. After that, after making your FASTA file, after getting the sequence, you make the FASTA file, and then you can open the MEGA 11 software. So now is that new version is there, maybe that MEGA is MEGA. So they are year by year wise, they improvise the software. So MEGA previously we had MEGA 7, 8, 9, 10. So like this way. So that new version is that MEGA 11. So software. So I'll show you that MEGA software. Yeah. So here's the molecular evolutionary and genetic analysis. This is the software out. Just when you open the software, you can get the these things, this window. So after opening the MEGA 11, so next step is the aligning the sequences. Alignment of the sequence means after we retrieve the sequences, we make the FASTA file, and then we have to do the multiple sequence alignment. So in multiple sequence alignment, we generally choose that you have to open a file session from the file menu, then sequence of file. That means whatever your file name is there saved in your desktop, you have to choose that file. So during like alignment, so there are two two methods are there. One is the cluster W and another one is the muscle. So these are the tool for alignment. Okay, so these two tool are developed by the Thompson, and this one is the Edgar 2004 2004B also they have developed. So either can be used muscles or cluster W. Both the tools are we can use for the multiple sequence alignment. So both the tools are can be available online also. You can do it also in online mode. So in the toolbar near the top of the window, cluster alignment is symbolized. So later I will show you that how that look like when we do the alignment. So if your sequence is DNA, you will choose two choices. Okay, suppose that your sequences is the DNA sequences, not the protein sequences. You can choose whether it is the aligned DNA or the aligned codons. So you know about the codons. So that codon means so that DNA is have on particular genome, the DNA sequences have some codons are present that code for a particular amino acids. So after choosing this, you have to click on the OK button to start the alignment process, so that within the few seconds, the alignment will be ready. When the alignment is complete, save the session and save as the same mass file. That mass file should be converted to the Omega, that means .make extension. That mass file should be .make extension. I'll show you the file later. Then after that, you have to do the estimate the tree. There are several widely used. So estimate the tree means that is the evolutionary model. Or you can say that there are different models are there for estimating the phylogenetic tree. Like for example, the neighbor joining UPGMA. So these are the basically there are so many tools are there or methods are there. So we can use it for the during the construction of the phylogenetic tree, like maximum likelihood. So basically we generally use that neighbor joining method and maximum likelihood method. For in this case, the neighbor joining method means how the organisms are related to one another. Neighbor joining basically we used for the like whether it is species level. For example, that in bacteria, so how the one bacteria is one bacteria is differ from the another bacteria. We generally use the neighbor joining method. Not only that, in case of species level, or we can also use it as a gene level also. So and then next step is that mega main window, choose open file, and then file menu, and save in the step 3 also. Save in step 3 means this one. So means after opening the mega files, you have to choose that mega your mega file. That means that when we multiple sequence alignment file. Okay, that step we have done this one, the multiple sequence alignment file. Okay, so and then we have to do we can choose that model, that is ML method, the maximum likelihood method. We can choose it and then click the complete button to start the or run your phylogenetic tree construction. So this is the main method to construct the phylogenetic tree. That is step 1, step 2, step 3, step 4, then step 4.1, 4.2. So these are the main procedures to construct the phylogenetic tree. So when we open the phylogenetic means mega software, when we open that phylogenetic tree construction, so this type of window will come. So I will show you that these are basically the parameters. Here's the phylogenetic reconstruction, maximum likelihood. The test of phylogeny is none. So here is that they have choose. They have choose another way. That means that they have choose that amino acids. So I'll show you that what I did. Yeah, this one you can see here. So I have done this way, like maximum likelihood method. Test of phylogeny is not. So there is no need to. These are the default parameters. There is no need to change. So if your sequence is the nucleotide, like DNA sequences, you have to change it here. Nucleotide sequence, whether it is your protein sequence or amino acid sequence, you should change it here. It's substitute type or substitutions type. Okay. So and then model is also is there. Model like Taimura name model, Kaimura model is there. So different type of model. So according to your experiment, you can choose it. Also that some parameters you can also maybe you can change it. Okay. So then uniform rates. So these are the some parameters. So the some parameters you can change it, or some parameters you can keep it as a constant, keep it as a that default parameters. So and then you can click it as a OK. So this is the main parameters of the during the phylogenetic construction. So here you can see here that phylogenetic construction. So that parameters we can change it. Some parameters, or some parameters we can't change it. So how we can retrieve the DNA sequences? For example, so suppose we don't have any DNA sequences, so you can directly go to the NCBI website. You can see here NCBI website. These are, so I have in that drop-down box, I have choose the nucleotide sequences like 16S ribosomal DNA of Staphylococcus aureus. I have choose that Staphylococcus and E. coli. So this Staphylococcus is the out group, and E. coli is the that inner group, or you can say as that our target organisms. Okay, E. coli. So I have choose that Staphylococcus is the out group, because the Staphylococcus is the gram-positive organism and E. coli is the gram-negative organisms. So then after that we can get the, you can see here also, we extract the sequence from that. We click it, we click it, and we can go to the FASTA sequences. FASTA sequences or FASTA file. So these FASTA sequences generally start with the greater than sign, and these are the file your organism name or the your sequence name. Okay, so like this way I have choose four E. coli and another outer groups, that is Staphylococcus aureus. And then next step is that I open the Omega software, okay, which is offline. You can download it from that download it from the online. Okay, so after that you can do that molecular evolution genetic is open it, and from the file you can go to the or retrieve the file. Okay, so from file you will get the import file. Okay, from import file you will get the sequences. Here you can see the E. coli AE12, AE2, then AY2, E. coli, and Staphylococcus. These are the four E. coli or bacteria bacterial sequences, for the E. coli sequences, and another is Staphylococcus aureus sequences. So these are sequences. After that you have to go to the alignment. You can see here, you can go to the alignment. So next step is alignment, and that when you get the alignment, you can see here also that it will be the yellow color. It turns into yellow color. When you select the sequence, it will be turns into yellow color. You can see here, I choose that you go to the alignment and you choose the muscle alignment, or you can choose as class tail W. Okay, so then after choosing it, so these are the default parameters. No need to change it. Just directly click on OK. Okay, after click on OK, so you can see it here that window will be appear. Okay, so these are the alignment sequences. Okay, so these are the alignment sequences. Whether the similarity, if there is a similarity is there, you can see it here. So that will be similar. If there is a dissimilarity is there, there will be gap is there. So okay, so that have a much that similarity and dissimilarity, it will take more time. I will not go to going through it. So just about that sequences. After click on OK, you will get the multiple sequence alignment. So that means that sequence have aligned among them sequence have aligned. Okay, after that, after sequence of alignment, you have to save the file, okay, as a mega format .meg. Okay, after save the file, and then after saving them, after you have done that multiple sequence alignment, you can do the construct the phylogenetic tree. Okay, so in the construct the phylogenetic tree, you can go to the phylogeny. You can see here in the software, you can go to the phylogeny. And after going to the phylogeny, you will see that here is the different construction or test maximum likelihood tree or construct test, neighbor zoning tree, minimum evolution tree, UPGMA tree. So these are the methods, test maximum parsimony tree. So these are the different methods we can use it for construction of the phylogenetic tree. So it depends on how you design your parameters or how design your experiments. It depends on your experiment. You can choose it like maximum likelihood method or neighbor zoning method. Basically, we generally use the maximum likelihood method and neighbor zoning method. After click on that maximum likelihood method, you'll see that that one window will appear. So you can see here. So you have to choose whether your sequences is the nucleotide sequence. That means that DNA sequences or the protein sequences or the amino acid sequences. Okay, so according to that, you have to select it. Okay, so another model or the method, that means the evolutionary model, which model you have to choose it. So that there is a different type of model is there, like Temura model, Kaimura model is there. So some other models also is there. You can choose it according to your experiment. Okay, so that red among sites, that should be that default by default. This one is no need to change. All these are by default. And then you can click on OK. Okay. So number of threads. The number of threads is like that. So number of sequences. Our sequences, I took that one is number of thread is 4, and another outer group I will choose that one is 1. Okay, so that some parameters we can change it, some parameters we can't change it. We can keep them as a default. Okay, then you have to click on the OK. After clicking on OK, so your phylogenetic tree is ready. So you can see it here, that phylogenetic tree. So you can see here that these E. coli organisms and this one is they have shared the common node. Okay, that means they have some similar characteristics because of that. So that E. coli AE2 and E. coli, so they share because they share the common ancestors because they have maximum similarity. Okay, so in compared to this one you can see it here, the E. coli AE1, 2. Okay, so have this group and this one have some similarities there. Okay, because of their similarity, they have some common node or the common ancestor. Okay, so E. coli, that AE1, 2, and E. coli and AK. This group and this one, some common ancestor is there because they have some similar characteristics is there. Okay, so another one is that you can see how they are, that one branch is another branch is how they are distantly related. Okay, so like E. coli AY2. So they that AY2 is some means what I said that is differ from these 3 organisms. Okay, they have some similarities there, but not so much. Okay, maybe there's some 70 to 80% are similarities there, but in that case like 90, you can say the 90% or 80% is similarity is that, but in that case below 80% similarity is that in case of AY2. So our outer group, in case of outer group, so this group and this group, you can see it here. So this group and this group have no similarity. Okay, because of there is no similarity, because of that this one is not totally connected with this group. Okay, so this is basically the phylogenetic tree. Okay, after that that means that one organism is how they are related to they are at the genomic level or the genetic level. Okay, so that means at the DNA level, you can say at the DNA level. So that how they are related, whether they have share some characteristics, share some some functions, whether they are phenotypically similar. Okay, whether genetically or phenotypically, maybe they have some similarities there. Because of their similarity, they are closely related, one organism to another organism. So you can see it here also. So these 4 organisms and this one. So how far related to this 4 organisms, the Staphylococcus aureus. So this is basically the interpretation of the phylogenetic tree, means how we can read or interpret the phylogenetic tree. So online tool like the iTool, also that is interactive tree of life. So you can go to the online, also you can use it as a construction of the phylogenetic tree. So you can see it here, different forms. You can see it that in the common node, if rooted or and branched. Whether the shape, topology, structure of tree of the tree is, it must have a common node, or if rooted or not, unrooted. So you can see it here. So suppose I have constructed this tree. Okay. So in the sidebar, you can toolbar, you can see a tree style. Okay, toggling, scaling of the tree, different type of tree is there. Okay, so you can here is the some also some in the toolbar, you can see also some format is there. You can use it for to design the whether it is bifurcating tree or the multifurcating tree, or the rooted tree or the unrooted tree. So you can, according to your design or experiment, you can choose it anyone. So you can see it here also, that how to interpret and read a phylogenetic tree. So here is that like A, B, C, D. Okay, it's a single type of tree, phylogenetic tree, but different forms. Okay, so these are the different forms of the tree. Here is the similar phylogenetic tree with the 3 different forms. You can see here A, B, C. There are 3 different forms of. So what is the application of phylogenetic tree? Basically, that evolutionary link between various organisms, evolutionary link between, suppose, the humans, how we can that related to our ancestors. Okay, like different like Neanderthals. Okay, so how we can relate it, or whether the how we evolve. Okay, so if we get the DNA sequences from like some hot springs, whether that sequence or our sequence have some similarities. Okay, or we can get some like got some sequences from the like fossils. Okay, whether it is plant fossils or animal fossils. Okay, or human fossils. So from there we extract the DNA and we do the phylogenetic tree construction. And how we see that after constructing the phylogenetic tree, we see that characteristic, whether we are related or share some characteristics. Is there any similarity is there, or not, or differences is there? So we can easily analyze it by through the phylogenetic tree. So not only that human being, also we can also find out the any evolutionary link between the organisms also, like microorganisms like bacteria, algae, fungi. Okay, so among them we can also identify, or we can say that how they are related among the among them, whether they have shared some common characteristics, whether they share the some some similar functions. Also, we can also classify organism and species in different taxa or different group also, like in taxa or group based on their DNA sequence and phenotypic similarities. So we can also divide the organisms also, like whether it is that some gram-positive bacteria or the gram-negative bacteria. Okay, some like in case of plants, also we can also use it in plants or animals also, whether it is like animal like cow, like buffalo, or like goat or deer, elephant. So among them, we can also classify them also, okay, based on their DNA sequences and some phenotypic characteristics. Okay, whether they have similar or different characteristics is there. Okay, so in addition to this, some like study the forces of evolution like, and characteristics of different organisms also. We can also like say that different, or in among the different organisms also we can find out the evolutions, how they are evolved. Okay, then also that we can also course of evolution and to classify species based on divergence of structures and functions, based on that different structures and functions, different type of functions. So among the organisms we can also classify them also. So this is the main applications of the phylogenetic tree construction, basically that evolutionary link between the or among the organisms, among the various organisms. So in conclusion, I can say that like construction means construction of the tree of life or phylogenetic tree is to relate the differentiate organism or species based on their phenotypic and genotypic characteristics. Okay, so we can say the phenotypic characteristics. Phenotypic is that not the genetically. Okay, so not is the based on the DNA sequences. So phenotypic means that invisible characters, some visible characteristics, like for example, like that in suppose animals. So we can say like animals have like 4 legs, 2 ears, 2 eye, okay, or whether the skin color. So based on that visible characteristics, we can also classify them also, or genotypic characteristics like genetic level also we can classify them. And there are so many different paid and free online software are also is there. So means some software is paid also is there in online. So you just submit your DNA sequences, they will do everything, okay, and give you the phylogenetic tree. Okay, so this is all about the construction of the phylogenetic tree. Basically, you need the DNA sequences. Not only the DNA sequences, you can get whether the protein sequences is there, so you can do the phylogenetic tree also, or amino acid sequences. So this is all about the phylogenetic tree, the how to construct it. So if you have any question, so you can ask it.
Students, if you have any question related to this topic, you can clear yourself with the doubts you have. Question asane Kaba? Or you can directly drop it in a chat box.
Yeah, you can share. If you don't want to tell, then you can share in the chat box.
The session is open for the Q&A. If you have any questions, our resource person will be delighted to make it clear. It was indeed a very informative presentation, Dr. Ahmad, and very interesting one.
Thank you, madam. Any question?
One question is from
the
Rashmi
time of
Butterfly.
divergence of two species by reading the phylogenetic tree.
I think we got one question in a chat box, isn't it?
Predict the time of divergence of two species by reading the phylogenetic tree. So basically that for the time of divergence, we can say that whether they have evolved in different times. Okay, So based on the genetic characteristics, we can predict the that divergence of time or the differences they have when they are evolve.
Thank you, sir.
So if you any question, then so regarding any confusion about the construction of phylogenetic tree, you can see here that basically this step you can follow this step. So basically that you have to select the particular target. Okay, so you have to particular target, and then PCR amplification, sequencing, you get the DNA sequence, and then you have to do the alignment. Okay, after doing the alignment, multiple sequence alignment. So multiple sequence alignments means more than one sequence. Okay, not only one. We can't do it that in one sequence we can't do the phylogenetic tree. So it should be like more than 2 sequences. Okay, 3, 4, 5, 6, 7, 8. Okay, more than 2 sequences. Okay, so that if the sequence is more than 2, that it should it say as a multiple sequence alignment. And then you choose the model, and you have to construct the tree. After construction, then you can evaluation or interpretation of the phylogenetic tree, or how means that whether the organism is related to each other, or they have share some similar characteristics, or maybe they have some share, they have some different characteristics. Okay, so some divergence is there in among them, or among the genes, among the particular genes or the group of organisms. Okay, I think there is no question.
Okay, we will proceed forward.
So another question, is there any minimum
question
length
is
of
there.
sequence?
Reaction over.
Okay, sequence for phylogenetic tree constructions. So there is no particular length, you can choose it. Okay, but if the sequence length is quite bigger, it will take more time. Okay, so it will maybe take like if we have the sequences is like 5,000 KB. Okay, more than 5,000 KB or 10,000 KB, it will take more time. We can use it, no issue. We can construct the tree also, but it will take time. But if the sequences is like 1.5 KB or 2 like 2 KB, so it can be easily in a short time we can construct the tree. If the sequences have like length is more than 2 KB or 3 KB, or like or the 5,000, 10,000, we can do it, no issue, that one, but it will take time.
Okay, we can't see any questions left here. Thank you for taking the questions and making it very easy and understandable way of how construction of the phylogenetic tree is being done. And yes, we are indeed very delighted and overwhelmed to have you with us today. We have almost come to an end of this session, 2-day invited lectures on biological science. I would like to express my heartfelt gratitude to Dr. Ramzan Ahmad for giving us his valuable time and for educating us with the knowledge on how the construction and the importance of the phylogenetic tree. It was indeed very informative session. I hope all our listeners have gained some insight into this importance of the phylogenetic tree. Now I would like to express my heartfelt gratitude to our principal sir, Dr. Sabyasachi Mahanta, Gorgaon College, for giving us the opportunity to organize this lecture series. Last but not the least, I would like to express my heartfelt gratitude to the esteemed faculty members and my dear participants for staying connected with us and for your active participation. Looking forward to more such inspiring and informative interactions in your futures with advanced methods. Thank you, everyone. Thank you, Dr. Ahmad. Thank you
Thank you,
so much.
madam. Thank you.
Thank you. With this, we would like to end this session.